Hey everybody, so welcome back. Today in chapter 3, we are going to be discussing nucleotides and nucleic acids. So after completing this lecture video and our two in-class activities, students should be able to describe the structural building blocks of DNA, as well as compare and contrast the building blocks of RNA and DNA. Additionally, we should be able to use the central dogma of molecular biology in order to predict protein sequence from DNA sequence, as well as predict potential malformations or mutations that may be seen in protein due to a mutation in the DNA sequence. Also, we will describe the forces that drive secondary structure of nucleic acids while comparing and contrasting the structures of DNA versus RNA. We'll also get a little bit of experience in assessing data in order to help us draw more general conclusions. So let's go ahead and get started.
Nucleic acids are comprised of chains of nucleotides, which are then comprised of nucleosides attached to phosphate groups. The most well-recognized function of nucleic acids is the storage and transfer of genetic information as DNA and RNA. However, nucleic acids can also serve structural and catalytic roles in the cell, are used in energy transfer and storage, intracellular signaling, can participate in oxidation reduction reactions, and play key roles in the metabolism facilitating biosynthetic reactions. When we deconstruct a nucleotide, we start first with the nitrogenous bases, such as adenine, guanine, thymine, cytosine, or uracil. Once a sugar is added, either ribose or deoxyribose, we then call it a nucleoside. If we add phosphate groups to the fifth carbon on the sugar of the nucleoside, then we call it a nucleotide. Nucleotides can exist as either triphosphates, diphosphates, or monophosphates, depending on the number of phosphate groups in the chain. There are a variety of bonds found in nucleic acids. Connecting the nitrogenous base and the sugar is a glycosidic bond, while connecting the phosphate group and the fifth carbon on the sugar is an ester bond. If we are connecting multiple nucleotides in a nucleic acid, we do so through a phosphodiester bond, where a second ester bond is formed between the preceding nucleotide's third sugar carbon and the next nucleotide's phosphate group. Hence why we often describe the sequence of a nucleic acid as running from the 5' to 3' end of the chain. In addition to the covalent bonds present in nucleic acids, we can also have hydrogen bonding. Hydrogen bonds exist between the nitrogenous bases of two strands of nucleic acids, often prescribed to DNA, although we can see base pairing and hydrogen bonding in non-helical RNA structures as well. Just as we have seen in the 3D protein structures, hydrogen bonding in nucleic acids is one of the major driving forces in nucleic acid folding and stabilization of 3D structure. Typically, bases pair up in a regular pattern in which guanine and cytosine form 3 hydrogen bonds and 2 are formed between adenine and thymine. We can categorize our nitrogenous bases as either purines or pyrimidines. Purines have 2 rings, where pyrimidines are formed by just 1 ring. Both are mixed atom rings, in which the ring is composed of both carbon and nitrogen atoms, hence the name nitrogenous. Each category follows their own numbering system regardless of the specific identity of the bases and their different functional groups. Adenine, which is also known as 6-aminopurine, has an amino group off of the 6th carbon, where guanine has its amino group off carbon 2 and a carbonyl off carbon 6, which can resonate to a hydroxyl group, hence the name 2-amino-6-hydroxypurine. Cytosine also has a resonating hydroxyl carbonyl and an additional amino group, while thymine has 2 hydroxy groups and a bulky methyl. Uracil, which is thiamine's replacement in RNA, is the same as thymine except it lacks the methyl. When we add a sugar to our purines, they are attached at the nitrogen in the ninth position of the ring, while pyrimidines attach at the nitrogen in the first position of the ring. It is the positioning of the sugars and the orientation of the different functional groups on the bases that then drive our differences in hydrogen bonding capabilities. Guanine has 2 donors and 1 acceptor, while cytosine complements guanine with its 2 hydrogen bond acceptors and 1 donor. Similarly, adenine has 1 donor and 1 acceptor to thymine or uracil's 1 acceptor and donor. The sugars found in nucleotides are either ribose or deoxyribose. Both are formed by the cyclic form of the 5-carbon long sugar ribose. Deoxyribose just happens to have lost its hydroxy group on the second carbon. In nucleic acid nomenclature, we often label the carbons in the sugars as prime to differentiate them from the carbons on the nitrogenous bases. While the loss of the 2' hydroxyl group alters the solubility of DNA slightly compared to RNA, it does increase the longevity of the molecule in the cellular environment and helps prevent degradation, which might be one of the reasons why the cell uses DNA for long-term genetic storage and RNA as the temporary messenger to deliver that information outside the nucleus. One nucleotide of special interest outside of nucleic acids is adenosine triphosphate. This molecule is used as the energy currency of the cell and is often coupled to inorganic reactions to help increase metabolic flux in the forward direction. The hydrolysis of the phosphodiester bonds between the phosphate groups of ATP creates highly stable products, releasing approximately 30 to 33 kilojoules per mole of energy per bond. In later chapters, we'll explore why energy is released during this reaction and how the cell controls the rate of that reaction in order to harness that energy when needed. The most well-recognized biomolecular structure throughout the world has to be the double helix of DNA. However, it wasn't until the mid-1950s that Watson and Crick predicted this structure based on X-ray data collected by Rosalind Franklin. While Erwin Chargaff found that the concentration of adenine and thymine was equal to each other and that guanine and cytosine were equal to each other in the early 1940s, scientists did not understand the base pairing potential predicted by Chargaff's data until that structure of DNA was solved. If you want to learn more about Rosalind Franklin and her lifelong dedication to science, I encourage you to watch the biographical video of her that we can find on our course webpage. The double helix is described as a right-handed anti-parallel helix, meaning one strand is running in the 3' to 5' direction, while the other is running in the 5' to 3' direction. The unique twisting of this helix also provides an alternating pattern of major and minor grooves. Indeed, different proteins, ligands, and drugs are designed to bind either the major or minor groove of DNA in order to drive or prevent certain processes such as replication and gene expression. While the B helix is considered the standard DNA helix and is the most common, there are other helical structures that exist in the cell and are driven largely by the nucleic acid composition. The A helix is more compressed with more nucleotides per turn and wider than B DNA and is typically considered dehydrated. The Z helix is actually a left-handed helix comprised of an alternating and repeating GC sequence that gives the helix an extended zigzag appearance. The human genome is composed of over 6 billion bases, which, if read out loud at a rate of 1 base per second, would only take about 100 years. Also, if you stretched out all of the DNA in your body, it could reach between you and the sun over 70 times. Needless to say, the cell has to get pretty creative in order to pack all of that genetic material into the cell that is only 100 micrometers wide. It does so first by supercoiling the helix and wrapping it around tiny proteins called histones, and then twisting that further to make chromatin. The chromatin is then packed very tightly into individual chromosomes. Unlike DNA, RNA exists primarily as a single-stranded structure that then allows it to take on some really interesting three-dimensional structures like protein with protein-like folding domains, stabilized by base pairing stems and loops. The protein-like folding properties of RNA actually enables RNA to behave as an enzyme at times with catalytic properties, such as we see in the ribosome. Indeed, some of the earliest prehistoric single-cell organisms on Earth are thought to have used RNA as opposed to peptides and proteins in their cell, and that RNA may have been the evolutionary precursor to proteins. Now, however, RNA is used to transfer the genetic information found in DNA through transcription to messenger RNA and translation to protein using tRNA and the ribosome. Additionally, RNA differs from DNA in that it is comprised of ribose versus deoxyribose for its sugar, and instead of using thiamine, RNA uses uracil. Both of these distinctions easily allows enzymes and other proteins in the cell to identify and utilize RNA differently from DNA. While the scope of this course is to not go into the finer details of DNA replication and transcription, it is important to recognize the implications of how mutations in genetic sequence can lead to mutations in protein that may lead to lethal or evolutionarily progressive changes. As a reminder, DNA replication is the copying of the genome prior to cell division. During replication, the two strands of the double helix are separated and copied such that two new strands are created, each with an old parent strand and a new or daughter copy. Therefore, mutations or changes introduced in the old parent strand will be replicated in the daughter strands every time a cell divides. Transcription is when we separate the DNA strand and make an RNA complement to the parent strand. The RNA is then displaced, allowing the DNA helix to reanneal, and the newly created messenger RNA strand will exit the nucleus. Once in the cytoplasm, the messenger RNA is then translated by the ribosome into protein. During translation, 3 nucleotide bases are read as a codon and code for specific transfer RNAs that recruit specific amino acids. Therefore, we can align specific 3-letter codons with specific amino acids and predict the amino acid sequence of a peptide from the parent DNA sequence. This connection between DNA sequence and protein sequence is ascribed as the central dogma of molecular biology. Again, if mutations occur to change even one nucleotide base, the change would call for a different codon and therefore a different amino acid. Additionally, mutations such as insertions and deletions could also cause the protein to gain or lose important amino acids. Because of the intimate relationship between a protein's primary sequence that drives three-dimensional structure and therefore function, mutations can be either lethal or trigger evolutionary change. When a proliferative cell undergoes DNA replication of over 6 billion bases at a rate ranging from every 20 minutes to 10 hours, mistakes occur. And while there are a number of proofreading and protective measures that the cell employs in order to minimize or correct these mistakes, some persist. Mutations can either be point mutations, swapping out one base for another, recombination, in which whole sections of a chromosome get swapped out for another, or transposition, where foreign DNA is inserted into a host genome. All of these can lead to malformations and misfunctions of proteins that lead to death or disease, but sometimes they lead to adaptations or improvements. Those types of mutations often drive natural selection and evolution over long periods of time as species become better prepared to survive against disease and predation. The Human Genome Project, which began in late 1990, had one goal: sequence the entire human genome. At that point, it took over $3 billion and 13 years of collaborative research to determine nearly 93% of the entire human genome of approximately 20 volunteers. In January of last year, 2022, the Guinness Book World Record was recently broken for sequencing an entire genome of one patient, and that established at just 5 hours and 2 minutes. This ultra rapid genome sequencing approach can revolutionize personalized medicine as we better understand how individuals' genomes vary and how that affects a person's response to disease and medicine. Interestingly, some of the things we learned from the Human Genome Project was that only 1.5% of the entire genome codes for proteins. The rest is considered non-coding DNA, or parts of the DNA that separates the protein coding regions. While the coding regions of DNA are of great interest as they eventually lead to proteins that govern cellular maintenance, function, and control, scientists have only just begun to truly appreciate the importance of that remaining 98% and the role that non-coding DNA plays in ensuring DNA integrity, regulation of expression, and cellular longevity. Indeed, it is the non-coding regions of DNA that we find some of these more unique non-helical structures that we'll discuss in more depth during our in-class activities. Until then, happy studying.